G'day, Dr. Carl, Shirtloads of Science, University of Sydney. And here's something weird. When you're about 10 years old, your brain, when you're just lying around quietly, uses up to 50% of all the energy you generate by eating food, blah, blah, blah. Oh, my God. And here to take us through it is Professor Dr. Sharna Jamadar. Is that the correct pronunciation? Dr. Sharna, I can call you Dr. Sharna?
Yep.
So you've been doing some research in this.
Yes. So we've been doing quite a bit of neuroscience research into how our brain uses its energy and how this changes across the lifespan. We're really excited to share it with you.
Yeah, so you've been using some new tools. I mean, for me, that's just astonishing. You've got this little brain, there's a few percent of your body weight, but in the case of a 10-year-old child, they're just lying there in the scanner, and it's using 50% of the energy. And I just realised the other day that talking is such a complicated thing to learn that you really don't get it right until you're about 12. It takes a long... Talking is such a complicated activity.
It really is.
And I've got a funny little video of my granddaughter trying to say happy birthday and can't concentrate on talking, and she falls over. She hasn't got enough processor cycles to be able to talk and walk at the same time. So you're a cognitive neuroscientist?
Yes, I am.
So what does that mean for say the geologists or the astrophysicists in the audience or the general public?
Yeah, so there's many branches of neuroscience, and so my form of neuroscience is trying to understand how our thoughts, feelings and behaviours emerge from both the structure and the function of our brain.
Wow.
So,
you pick a big one, don't you? Wow.
Yeah, just
That's
a little
very
problem.
difficult one, yeah.
Yeah, and so we use biomedical imaging, so things like MRI and PET imaging, and get people to do different tasks in the scanner, and then we get their data and we look to see how their cognition, their psychology is related to their brain function. And
you've made some very amazing stuff I had no idea of, but just as a bit of a background first. So if you do an X-ray, all you just sort of get is a straight backwards, forwards shadow of the whole brain. If you do a CAT scan, you can do more detailed imaging. But the trouble is you've got this stupid thick skull in the way.
Yeah.
So if you do an MRI scan, the skull is no longer visible, and so you can get much more detailed info. Is that correct?
Yeah, that's right. So MRI gives us really exquisite what we call spatial resolution, so we can get to the sub-millimetre level.
Well, sub-millimetre?
Yeah, exactly.
So if I draw a line on paper, that's probably 0.5 millimetre wide. You can get how much, like a 0.5 millimetre?
Yeah, about that, yeah. So we can start to get to some of the cortical columns and see how the different columns are organised within the cortex.
What are these cortical columns?
Yep, so columns are basically characterised by different morphology or types of neurons that you have throughout your cortex. And so our brain tissue is comprised of lots and lots of different types of cells, and you can start to differentiate those cell types with the MRI.
I was just reading that late last year, after a 5-year study, they found over 3,000 different types of cell types in the brain.
Yeah,
which was just astonishing because before that we thought there were like a couple of dozen.
Yeah.
And they spent 5 years doing this. Okay, so you're a cognitive neuroscientist, and you've made some amazing findings which totally blew me out about the glucose metabolism in the brain and how it varies. Okay, so you can look at the anatomy, and then you're saying using PET, is that positron emission tomography?
Yep.
And so you're using that to molecular imaging. Can you tell me what you're looking for with a PET? And you blend these together. Yeah.
So we use a really sophisticated machine called a simultaneous PET MR scanner.
You can do both at the same time?
Do both at the same time. And so this is really, really powerful because we can get really exquisite anatomical information from the MRI. But then from the PET, we also get really beautiful molecular information. And so what that means is that we can track a lot of the physiological processes in the brain that we couldn't do with the MRI. And so one thing that I do is look at glucose metabolism, so look at the way that the brain is using energy. And to do that, we inject a radiotracer into the body. And so this is a small pharmaceutical with a small amount of radiation.
Is it glucose?
It is. It's a glucose analogue.
So it's essentially radioactively labelled glucose in effect.
Exactly. Yeah.
So would the brain break it down and use it for energy in the same way it uses regular glucose?
Up until a point. So when you first inject it, the body thinks, okay, this is glucose, and it just starts to move about your body in the same way that glucose would. It enters the brain in the same way that the glucose would.
It will cross the blood-brain barrier?
It will do, yep, right. In the same way. And then it will start to be metabolized by the cells in your brain. And then at a certain point, the body will realize, oh, this actually isn't glucose, this is something else. And then it will stop. And when it stops, the radiotracer stays trapped where it is and just kind of sits there and passively decays. So it emits a positron every now and again.
And a positron is a positive electron.
Yep.
Okay.
And then, so while you're lying in the PET camera, we detect those positrons, and so that allows us to determine where the brain was using the glucose.
So by using this combination technique, you get, firstly, very detailed scanning of the anatomy of the brain, and you're saying to maybe half a millimetre or a tenth or half? Half. And then also you're getting good information on what metabolism of glucose has been happening.
Yep.
And then you mentioned here FDG.
Yeah.
Is that the chemical?
That's the chemical. So that's what we're injecting you
with.
What
does FDG stand for?
So it's a bit of a mouthful. It stands for fluorodeoxyglucose.
Fluoro, so it's got some of the fluorine chemical, which is a very corrosive gas, and which they use to etch glass with.
Oh.
And one of the very few things that it won't attack, it'll attack anything, is beeswax.
Oh.
So you get a big sheet of glass and you put a bit of beeswax and you make your little pattern and it'll attack where the little pattern is. So it's
deoxyglucose.
So you've removed a little bit of oxygen and then it's glucose.
Yep.
So you do have a regular glucose there.
Yeah, exactly right.
So it can be burnt for energy.
Yeah. It tricks the body. So for a while, it does think that it's actually glucose, and so it just gets sucked up by anything that's looking for glucose for energy. Okay,
so just before we leave talking about the tools that you use, so this is like a regular MRI scanner in the sense that you're not allowed to take a bicycle chain in there or any of that other stupid sort of metal.
Exactly.
Right, okay.
So you have to be really careful, and we make sure that you have no implants in your body that would be likely unhappy with entering such a strong magnetic field.
Right. Okay. So, hang on, one more thing. Can you tell our audience the blood-brain barrier? Because reading one of the papers that you authored, for the first time I understood the blood-brain barrier, which I didn't in medical school. So just can you explain for us what is the blood-brain barrier and why do we need it?
Yeah, so the blood-brain barrier is there to control and regulate what enters your brain, so it stops stops nasties from getting in that we don't really want to get in. Of course, some things can still get in. It's not perfect.
So I'm guessing you want glucose to be able to get in.
We want glucose to be able to get in.
But
exactly.
Bacteria are probably blocked?
Well, some is, yes, and they can evolve to cross the blood-brain barrier, but blood-brain barrier is basically comprised of like all these channels, and the idea is that these channels can open and close to preferentially let certain molecules in.
Okay, so it's like if you imagine you've got a blood vessel, like a tiny artery, and it's got kind of a pipe around it, and then in that pipe, and you said it was made of astrocyte feet and endothelium and basal lamina. Look up in Wikipedia. So channels can open in this and they will let through glucose?
Yeah, exactly.
So the blood-brain barrier will let glucose and similar molecules through it?
Exactly, yeah.
Right,
yeah.
okay. So that's a bit of a background for the blood-brain barrier. So it's always astonished me that the brain is so light and uses up so much energy.
That's right, yeah. So when you're an adult kind of walking around in your 20s, 30s, 40s and 50s, your brain is about 2% of your body's weight, but it's using about 20% of its energy.
That's huge.
It is. It's about 10 times as much that what you'd expect just based on its volume alone.
And when we mentioned before, 10 years old, it's using about, and you're lying quietly in the scanner, it's using 50%.
Yeah.
It's laying down, what is it doing?
So it's a little bit of a mystery about exactly what is happening.
Because it's the brain and we don't understand.
Exactly, yeah. So you might think that it might be related to puberty, for example, because like after, when you're like 11, 12, 13 or so, your kind of growth rate really like accelerates throughout the rest of your body. But intriguingly, it seems to actually not be related to puberty. It seems to be something else. And what is believed at the moment is that it's related to this process of synaptic proliferation and pruning.
Okay, can you explain that for the metallurgists in the audience?
So you might think that all the neurons in your brain are actually like physically touching each other, and that's the way that information kind of transfers between different cells. But that's actually not the case. So they're actually just separated by these tiny little gaps called synapses, and then molecules are released between the synapses. And
little chemicals?
Yeah.
So you have an electrical thing happening along the nerve, and then when it gets to the end of the nerve, it says, don't care about electricity, now I'm going to talk to the next nerve with chemicals.
Exactly, yeah. And so you've got these things here called synapses, and when you're 10 years old, you're building a lot of these synapses. So you're making connections where they weren't there before. That takes a lot of energy, but on the flip side, what's happening when you're 10 years old is that you're starting to prune away connections that are no longer needed. And this pruning process is really, really important, because if you don't prune away enough, or if you prune away too much, then your brain is not very efficiently kind of wired. And so there's this simultaneous process of both building the connections as well as taking away connections that are no longer really necessary. And that is kind of peaking when you're 10 years old. And so that's why we think the brain is kind of like really just sucking up all that energy at that time.
When I read the paper about the 3,000 different types of nerve cells, it pointed out that on average, each nerve cell in the brain has not one or two connections like a transistor, but 1,500? Jeez.
Yeah.
There's so many things saying, do this, don't do that.
Exactly.
So, like, I'm guessing long gone are the days when you said, this bit of the brain did only that. No, it's more complicated.
Yeah.
And so the energy, can you talk a bit about how glucose goes into ATP and why this is important for the brain?
Sure.
Did you try to monitor ATP as well?
Not directly, so it can be quite difficult to do that with just through what we call macroscale neuroimaging, so where we just get people walking around in everyday life off the street and come into our scanner. So we kind of indirectly measure that through our PET imaging. And so when the brain needs energy, it'll metabolize this glucose, and when it metabolizes the glucose, it generates between 30 and 32 molecules of this ATP, so this adenosine triphosphate. And so ATP is called the energy currency of the body and the brain. And so that sounds kind of obscure, but basically any cell in your body will accept ATP as an energy source. And so it's kind of like the euro of the European Union. Everybody will accept it.
It's like the cash.
Yeah, exactly. Yeah.
It used to be cash was legal tender. I suddenly realised that's a bad example. So if you want to blink an eyelid, move a muscle, shift blood, shift air, clean stuff out of your kidneys, ATP has to be consumed and turned from triphosphate into diphosphate. And then that releases a lot of energy. And so one molecule of glucose, which is a bunch of carbon atoms in a ring, will give you 30-ish molecules of ATP.
Yeah, that's right.
We won't even talk about ketones, that's a whole different thing.
Yeah,
okay.
yeah.
So how does it change as you get older? So you start off young, and we've got a little grandkid. It's amazing how just in the first few weeks, the hand movements go from very spasmodic to less spasmodic and more controlled.
Yeah.
And you can see things changing virtually on a day by day, and you're thinking, God, that little brain is working hard.
Yeah.
But what happens when you get past your 20s, say into your 70s, 80s?
Yeah. So naturally, as we age, our brains consume less energy. And so by the time you're 80 years old, your brain's consuming about 3/4 as much energy that it was consuming when it was 20. And so we think about this as being a smaller energy budget for older people. And so what this means is that not only do you have less energy to kind of like drive stuff, or your cognitive processes, or your thoughts and feelings, behaviours. But you also have less energy to do other physiological things, so clear out toxins that might be associated with Alzheimer's disease. It also means there's less there to control inflammatory responses as well, and also to synthesise your neurotransmitters like dopamine and serotonin and so forth.
Oh, they have to be made?
Yeah, they do, yeah.
Of course, they don't come from nowhere. They're not in your food. They have to be manufactured.
Yeah, and so glucose is important for that as well. So you've kind of got this smaller energy budget that you need to basically drive everything else that you've been doing your whole life.
But hopefully evolution is kind to you and makes things a little bit more efficient for you as you get older, or not?
Not quite.
No? Less efficient?
Yeah,
oh my
yeah.
God. So evolution doesn't care about you.
Yeah.
Past your 20s, you're over the hill kind of.
Yeah.
Wow. So I've been reading some of your papers, and you kind of point out that this brain is this kilogram and a bit of stuff, but somehow out of it comes consciousness and emotion and the Michelangelo paintings and Beethoven and Savage Garden and God knows what.
Yeah.
How does that happen?
So our brain is really metabolically hungry, so it's sucking up heaps and heaps of energy, so about 20% of your body's energy, but it's also remarkably energy efficient. And so it's running on about 17 watts of power, which is incredible considering that it's currently more intelligent than AI, but they're talking about building these AI data centers next to nuclear power plants because they're just so energy intensive. And yet our brain is running on less than an old school incandescent light bulb.
Wow.
So it's kind of evolved to use its energy really efficiently. And so the way that it does that is that it's both organised efficiently, but it also uses the energy efficiently as well. So in terms of organisation, you might think, well, all parts of your brain are kind of interacting with each other all the time. So therefore, maybe they're just all connected to each other in some way. But it turns out that it's not, because if you want to send... It's not, no. So if you want to send information, say, from one side of your brain to the other, then that's really energy intensive. So that costs you about 35 times more energy than what it costs you to just send information, say, between two brain regions that are side by side.
That are on the same half.
Exactly, yeah.
Really?
Yeah. And so your brain preferentially is connected to regions that are kind of closest to it and is also involved in similar kind of like cognitive tasks. So say like the attention centres are kind of connected to each other, for example. And then what happens is rather than actually sending information across the whole network kind of haphazardly, it's organised a little bit like our telephone networks or domestic airline networks where the information transfers through certain hub regions. So say, for example, if I wanted to fly from Melbourne to Cairns, a lot of the time I've got to go through Sydney because Sydney is the hub region.
So hub and spoking.
Exactly, yeah. And so those hub regions use more energy than non-hub regions. And it turns out there's a really energy efficient way to organise a network. And so we organise our man-made or human-made networks in using these principles, but our brains have kind of like evolved to use this principle on its own. So that's one way that we conserve energy. The other way that we conserve energy is by only firing our neurons when we need to. We have these models of the world in our brain and we fire whenever something violates that model. So you might, for example, be sitting in a room and there's this really, really annoying beeping noise going on, and over time you won't really notice it anymore because the beeping is just kind of consistent, it's not changing, and your brain is like, okay, this is what my world looks like, it's got this really annoying noise, but I'm not going to pay too much attention to it. But when the noise changes and maybe changes frequency or pitch or something like that, then your brain is like, oh, hang on a minute, the world is not what I expect it to be, and so therefore I need to pay attention and see what's happening. And so these principles of predictive coding, where you're just kind of firing only whenever something violates your expectancy, also helps conserve your energy as well.
Wow. Just as a total diversion before we get back, is there any point in trying to grow, with regard to AI, brain organoids and then use those collections of brain cells to do efficient thinking for us and not have to worry about things like moving your arms and legs.
It's funny that you say that because a close colleague of mine is literally just doing that.
Really?
Yes, he is. And
following the old rule of do not reinvent the wheel. So a colleague of yours is actually thinking about, can you just explain to me a bit more?
Yeah, so they've basically grown, as you said, these organoids are based on neurons. They've taught them how to play Pong, the old school computer game.
Was that the one I read about in the paper?
Yeah, yeah.
Oh, my God.
Yeah,
one of your colleagues.
Yeah.
So how big are these organoids? Like, are they about a kilogram or a gram or...
I'm not quite sure.
Something small.
Yeah, something small.
And they can play Pong.
Yeah.
Where you just got to move the bat to reflect the ball back across to the other side.
Exactly, yeah.
Okay.
And what's fascinating about that actually is that the human neurons are better at playing the game than mouse neurons.
What? So we humans, our neurons can play Pong. Okay, right. Okay, I'm just trying to absorb that. Okay, thank you. Yeah. Wow, isn't that weird?
Yeah.
With this whole energy thing, now the variability. Now, I had no idea about this. So this is one of the things you've just come across recently. There's not just the size of the energy budget, how much glucose you're burning, but how variable it is, how you can ramp from up to down. And can you talk about that?
Yeah, that's right. So our research is really looking at this kind of scalability of glucose metabolism in the brain. So in other words, the ability to up and down regulate it on demand.
Oh, so scalability means how much you can vary it up and down. Yeah. I didn't think about that. I just thought it just sat there going 20%, 20%, 20%, but it's scalable.
Yeah, exactly. And so when you think about it, it does make sense, right? When you're doing a hard task and your neurons are kind of firing away and sucking up lots of energy, they're using more glucose. But in general, neuroscientists have found it difficult to actually measure the variability in real time. So now that we can measure this, what we're finding is that the size of the energy budget is important to a certain extent. But what is most important in terms of driving your cognition is actually your ability to upregulate it on demand. And so it's the size of the window within which you can upregulate it up and down, but also the complexity within which you actually use that glucose as well. So that's kind of a measure from physics called entropy. So the complexity of that signal is also really important for driving your cognition. And so what we find is that, for example, in healthy older adults, that kind of window within which you can vary your glucose metabolism starts to get a little bit narrower. So you can't really flexibly adapt your glucose system as well as when you were younger.
So firstly, as you get older, you are using a little bit less glucose. Secondly, the variability, the difference between the base level and how much you can use. But then you threw in another thing at me, complexity.
The idea is that a complex brain signal can carry more information. The idea is that if you've got a more complex signal, say transferring information from your frontal cortex to your visual cortex, that's a more efficient way of encoding information in your brain. And so what we find is that the way in which we use glucose, the complexity is also really, really important because it means it can convey more information, and that also changes in healthy ageing as well.
Okay, so glucose is used by the brain, and I'm guessing an important chemical from my endocrinology is insulin from your pancreas. So what controls the glucose levels?
It's really interesting because the brain both is reliant on glucose as its fuel, but also controls how much fuel is available. So you have to see that if it's both reliant but also controlling it, that means that we have to maintain the amount of glucose in our blood in quite a fine balance. And if anything kind of disrupts that balance, then your brain can start to deteriorate and kind of cry out for some more fuel. And so, as you said, insulin is really, really important for this. And so a small part of your brain called the hypothalamus will send signals to the rest of the organs in your body, so say like the liver and the pancreas, but also your tissue as well. And it will kind of signal and say, hey, I need a bit more glucose, or actually, do you know what, I've got enough glucose at the moment, kind of stop producing it, and so forth.
There's various things that happen to the brain with time. neurodegenerative diseases, amyotrophic lateral sclerosis, Huntington's, Alzheimer's, etc. Is the role of glucose important in this, and is the role of glucose in your early childhood years possibly related to something in later life?
Yeah. There's an increasing recognition that the way that your brain is using glucose is a really important factor in determining whether or not you'll go on to develop particular disorders. As you said, Alzheimer's disease really closely linked to disruptions in glucose metabolism.
It is?
It is. But then also other conditions like schizophrenia and major depression and so forth also seem to be related to kind of subtle metabolic disturbances. And so for decades we've kind of been trying to figure out kind of what is the causative factor. And it turns out that at least in the drug discovery space, a lot of people are starting to focus on early disruption of the metabolic system.
Early? How early?
Well, again, not 100% sure. Midlife is always like a really important time when you're talking about disorders of ageing. But, we know that, for example, that if you have quite acute disruption to the supply of glucose to your brain, that this can be really catastrophic. If you think about metabolic disorders like diabetes, for example, you can go into a diabetic coma and things like that. So it can really disrupt your consciousness. And so that's what happens if you have like a really kind of like abrupt disruption. But if you have a more subtle disruption that kind of is chronic, it goes over several years, that fine balance between your brain and body and its fuel source starts to become disrupted, and over time you just won't have enough fuel for all of the processes that your brain needs.
Wow. Like they do say from ancient times, healthy mind in a healthy body, and a part of the healthy body is not just the exercise but the diet, which you don't get if you eat takeaway food every day, as opposed to my favourite, which is steamed vegetables with steamed soybeans and a bit of tahini. So, Dr. Sharna, would you describe yourself as a cognitive neuroscientist? That's okay.
Yes.
But how did you get here? Like starting off in baby jail, primary school, high school. So what was your educational pathway and career pathways to get here?
So I did an undergraduate degree in psychology. I grew up actually not far from here, a couple of hours north of Sydney.
Yep.
Went to university at the University of Newcastle.
I love New.
Yeah, did psychology there, fully intending to kind of finish up and be a clinical psychologist. And then as I was completing my honours year, which is a year of intensive research at the end of your degree, I just absolutely loved the research, and I was so inspired by a number of the people around me. And so I thought, you know what? I want to do what they do. I want to be a scientist just like them. And so I kind of shifted what I was doing and enrolled in a PhD. And so that was quite neuroscience focused, trying to use brain imaging to understand cognition.
So what was your honours thesis work?
My honours thesis was very different. So it was actually in vision in guinea pigs and trying to reverse myopia. And then the PhD that I did actually was using two imaging technologies, so EEG, so that's the brain waves. The
little squiggles up and down, from a bunch of electrodes around the skull,
Exactly, and fMRI, so
There's functional MRI where you have radioactively labelled something?
No radioactive.
No radioactive.
So
you don't need radioactive?
No, you don't.
Really?
So we basically map what the brain is doing by looking at changes in blood oxygenation.
Oh, you can look at that directly.
As
well as anatomy, you can look at brain. I had no idea. Wow, okay.
So every time your brain is firing, it's using both glucose and oxygen. And so the fMRI will allow us to look at the changes in the oxygenation.
You were doing that in your PhD? And what did you find?
Well, we found that the way in which the brain uses the oxygen and the electrical potentials differ between people with schizophrenia and people without schizophrenia.
Really? There's a difference?
When they're doing particular cognitive tasks, so planning actions and things like that.
So if they're trying to say, I want to go from here to there, I've got to take a bus, a train, walk, taxi, blah, 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 there's a difference between a person with schizophrenia and a person not. not when they're in a schizophrenic florid stage, but just when they're regular thinking. There's a difference.
Yeah, exactly.
Wow.
Yeah, so the way that they plan all those actions is different to the way that a healthy individual would.
Wow. So Dr. Sharna and Professor at Monash, I'm guessing you have PhD students?
Yes.
So how can people follow you and become your student, either through social media or just the regular boring university website?
Yeah, that's right. So you can follow me on Bluesky.
Bluesky?
That's right. And I also use...
And what's your handle on Bluesky?
At sharnajamadar.bluesky.
So SHARN for November A, and no dot, and then straight into JAM for Mike, ADAR.
Yep.
Bluesky,
Yep, that's right. I also have a website, so sharnajamadar.com, where we post all of our papers and things like that. And you can also contact me at my Monash email address, which is sharna.jamadar@monash.edu.
Oh, wow. Oh, look, I hope that somebody will find a nice career working with you in the future.
Thank you very much.
Thank you for coming in today.
Thank you.
Shirtloads of Science is washed, spun and aired by the University of Sydney.